Okay, so first we're going to do the chicken and egg problem. We've already shown that if DNA is long, actually I think that's a homework problem. Has anybody done the homework problem? Because it's due Thursday. If DNA is fairly short, let's say 10 base pairs, the probability of it coming unwinding is really low or really high? Really high. And as the DNA gets longer, the probability of it opening tends to decrease. Okay. And that's because we know that the base pairs are hydrogen bonding. Okay, so as we know, we have this incredibly long DNA in our body. So the chances of it coming unzipped is like about 0 due to like thermal energy. So how do we do that? Because our DNA does need to unzip, yeah? So how do we do it? Come on. Proteins. Proteins. You have a protein that comes and, let's say, acts as essentially a wedge and comes and goes and breaks apart the DNA. By the way, where does it get the energy for that? ATP, and we'll go through what ATP is. But ATP contains a little burst of energy, takes that energy and then goes through. Okay, so it needs proteins to catalyze the reaction, and now you know what that means, that initially it's zipped. It's really high energy to make it over this transition state to be unzipped. Instead, so what you do is you add a protein, feed it some ATP, and essentially it lowers the energy such that it can happen. Okay, but it needs proteins in order to unzip. But how does it get the proteins? You know, DNA must go to RNA, must go to protein. So how does it make the protein initially? Okay. Well, you're very close. That's right. This is basically the fundamental problem. And the answer is, well, it's a little bit long-winded. What it is DNA doesn't simply go to RNA, which goes to proteins, or DNA goes to DNA involving replication, because what DNA first does, it makes what is called pre-RNA, pre-mRNA. Sometimes it's called nuclear RNA. And that indeed is exactly a copy of the DNA. It starts at a start codon, ends at a stop codon. Then that pre-mRNA or nuclear RNA is made. It then leaves the nucleus and becomes RNA. What's the difference between pre-mRNA and mRNA? It turns out that there is, in DNA, for example, here, you have what are called exons and introns. Exons are truly expressed. That is, they end up making an mRNA which goes to a protein. The introns here are so-called dead weight. And so what happens, you have the nuclear RNA or the pre-mRNA, then you do the processing where these guys are cut out from the inside. These are stuck together again, and then you get a protein from that. And it turns out that this process is really, really important, and it is ubiquitous. In fact, something like 90% of DNA, it makes a pre-mRNA and then makes an mRNA, and they're not the same. So for example, a hemoglobin molecule. This is the DNA sequence. It starts at a start codon and ends at a stop codon. It makes that. The pre-mRNA goes outside, and then actually what is happening is you have this, and you have this, and you have this. And everything else is cut out. So it turns out that this adds a tremendous amount of complexity to ourselves. And that I told you one gene codes for one polypeptide, which maybe a couple of polypeptides come together to make a protein. In fact, one gene can form many proteins. So in fact, you have an exon here, an exon here, and an exon over there. And they can combine, eliminating both, or rather, yeah. Or this can combine with this, or this, or this. And it turns out that, again, this is ubiquitous in that, for example, this Down syndrome cell adhesion molecule is 38,000 potential mRNAs. How do you get that, a number like that? How many introns would they have to be? Okay, well, I'm actually not sure. Good. Why don't you spend 2 minutes saying how many introns about there must be for you to get 38,000 mRNAs. Go ahead, talk to your nearest neighbor. Yeah.
So I don't have any biology at all, but this is totally cool. But like, has any of this ever been like distilled into like technology, and like uses like either like a compression algorithm or like security features? This whole whatever, 28,000 isn't that big of a number, but like if you could increase the magnitude by a couple, you probably have like a good key for some cryptology system. Yeah, like just the way it works.
I don't know. I don't know.
That sounds like a big
Could we deal with it afterwards? Yeah.
So, in this example, it always goes like in order or whatever. If you flip like orange, for example, like in the second one where it's blue orange, can you do that, or can you not ever flip the sides like that? Do they have to be in the order as they go up in the genome?
I don't know. Honestly,
I would think
I-
you can, but
I don't know. Okay,
They can.
so what is the general idea behind, you get one pre-mRNA and then you know, and this might be an exon, I don't know. This might be an intron, this might be, this might be. So how do you solve it?
Can there be two introns together?
Excuse me?
Can there be two introns together? Wouldn't it just be an intron?
Yes.
Can you repeat the question again? Were you asking how many ways there are to slice out an intron?
Yes. In effect, that is because you could have exon 1 and then exon 2, and let's say intron, an exon, and you could have exon 3. So you've cut out all of the introns, although maybe you can have exon 1, intron 1, exon 2, exon 3, etc.
Can we have introns as well in RNA?
Yes, absolutely. Yes.
But they won't go to the protein.
Well, in this case, you're saying, can we have the exon course? This is the mRNA. Can you have an intron in the mRNA? Yes?
Yeah, for example, the sex determination is determined by whether you have that intron.
There you go. I like when a person has the answer. So say it again, say it loud.
The mechanism of sex determination is dictated by whether you have one specific intron in one of the genes. So you can, when long short, you can have
Intron,
in the mRNA.
right?
Does it code to protein intron as well?
Yeah, we
Yes.
can transmit it.
So it will represent.
Okay, yes.
So I guess I'm a bit confused because if the intron is like determining things like sex and everything, then isn't it then being expressed? How is it still an intron if it determines?
In this sense, it's not, because the intron becomes part of the mRNA, and the mRNA then codes for a protein, and these codes for particular amino acids. It is an intron in that it can be cut out, and, for example, over here. Yes.
So like regarding to what you said like there's a specific gene that they just like do for sex determination and stuff. So like technically the definition of genes, like you have introns in your genes, and then like you copy those genes to pre-mRNA and you chop out the introns and make it mRNA. So I mean like, so based on this flow, you shouldn't have any introns in your mRNA, but you can have like as many introns as you want in your genes. So that's not the correlation that
Yeah. In your DNA, you have a string of bases, and some will be exons, meaning they will be expressed as mRNA, and some will be introns. The introns sometimes will be expressed, and other times they will be cut out. Okay, yes. Okay, yeah.
Just to follow up his questions, since you said that introns are basically just junk, information.
Well, why do we have them?
No. Why are they included in the transcript? It's
Well,
like again, just like him. My preconception was that all introns must go.
No, at least that is not correct. They sometimes go. Okay. Okay, yeah, one more question, go ahead.
Similar to what he said, I mean, the DNA replicates, so it might take the intron to the next cell, and that might split some of the exons later on or something, and that might lead to sexual determination, but I don't, doesn't intron actually just define something that doesn't go to the RNA, and just stays as a pre,
Okay, I'm not sure in terms of I believe the intron is effectively surrounded by a particular sequence. And what it says is, it says sometimes eliminate that sequence, other times don't. And why it does that, I don't know.
but then all exons go through? That's not true either, right?
All exons go through, yes.
All of them.
All of them, yeah.
I mean, if it's sometimes, then introns is the same thing.
Yeah.
Sometimes.
So one of the classical examples of this is for how we recognize invading pathogens. So our antibodies are one of these big, long proteins, all expressed as one gene, but depending on how they get spliced, they will recognize different antigens. So sometimes the exons are actually kept in because, they make the heavy chain or the light chain, I forget which chain is which of the antibody, but you need the stuff on the tips to be different every time. So sometimes those exons are kept, and other times they're spliced out.
So then, the definition of the exons and the introns, that seems a little funky in terms.
So my understanding is that the introns are always taken out. It's the exons that may or may not, depending on how the splicing takes place.
Okay, but that seems to be contradictory what he's saying, in that sometimes the introns are taken out in the sex determination scheme, and sometimes they aren't.
Well, biology is weird.
Actually, that he's made there's some confusion, and I need to check on this. But it is something that whenever you have a general rule, in physics, you have 3 laws of mechanics and 4 laws of E&M. In quantum mechanics, you have, 1 law, the Schrodinger equation. It's true. In biology, what you have is you try to make these general laws, and you find that they're not universal. In fact, this is one situation going from DNA to RNA to proteins, which people thought, this was a central dogma of molecular biology, but actually it's not. Okay, last comment.
Did you mean that introns are always spliced out, but not all introns are spliced out? But in the case of exons, they're sometimes spliced out. So the difference between introns and exons would be that introns are always spliced out, not all, but some, but exons it depends.
I think that's basically saying the same thing.
No, not that all of them are spliced out, but every time there is a replication, that introns are definitely removed, some of them or all.
Some or all of the introns, yes. And then the problem with exons, the question is, do they always code? Are exons ever cut out? Do you know, Josh?
I mean, I only know as much as I learned in biochemistry, and that was a while ago.
Okay. So I have to go and do a little bit of homework. If anybody wants to help me out, that would be great. So can we continue, please? Okay. So, oh, wait. We never got to our original problem of how do we get 38,000? So how do we do it in terms of, here we have 3 exons, 2 introns, and we're going and we can form 4 mRNAs. So let's take this and try to generalize it. So what did you conclude? What did you get any sort of generalization? Yes?
Factorial.
Okay, it sounds like there's some factorial. I think that's right. What?
If we assume that all the introns are spliced out and if there are n exons in the system, then again one more assumption that they can combine in any order.
Okay, yeah, that is another question. For example, can they in fact combine in any order? Like this, could it actually form a white and then a blue?
Yeah.
I don't think so. And he says no.
The spliceosome won't let you do that. So the thing that actually does the splicing, there's
Yeah.
a spliceosome, and it can only take one thing to the other thing, so it's always in order.
Okay, so we have to account for that. Okay, this is going to be non-trivial. Good thing I didn't try to do this. Oh, wait.
Well, so it's still factorial.
Oh yeah, it's still factorial, but it's going to be some interesting combination. Gee, interesting. What does that mean? It means you're going to have to do it for homework. You didn't find that funny. Oh well. So I have to figure it out, and I have to make it up.
Small n, please.
What's that?
Do it for a small number of n.
We'll see. Okay. Anyways, so here you have 79 introns, 14,000 nucleotides, etc. So the question is, why are you bothering to have all these introns? Also, and how do you recognize them? Turns out that it'll be very important in regulation of the gene in terms of whether the gene is actually expressed. Okay. Yes?
Did we answer that question? Where does that splicing for protein come from?
No.
Okay.
But let's assume that there's some protein that comes around that does the splicing. And in fact, I'll come to that on the next one, because it turns out the answer to that is really critical. Okay, but this explains why we're more complex than a tiny worm. That is, this worm called C. elegans, which is about 1 millimeter long. It's been the topic in terms of understanding it. It turns out that the little worm has 19,735 genes. And that is approximately equal to 21,000, which is the amount of genes that you have. But in fact, a lot of your genes are alternately spliced because they have a lot of these introns. So in fact, the idea of one gene going to one protein is not really true anymore. So in fact, we have a tremendous number of genes because we have so many introns, and therefore we can produce an enormous variety of proteins. Okay. Now, how are the introns and exons made? It turns out that they're made by the mRNA coming into what's called the spliceosome, which is actually a fairly new complex. It turns out that the spliceosome is made of not only proteins, but actually made of RNA. So the majority are cut by the spliceosome. However, a minority self-cleave. The fact that they self-cleave means the implications are huge, that RNA can be genetic information, but also it can be a catalyst. It'll go and cut either itself or other RNAs. So let's understand, well, first, why this is such a big deal. That is, RNA solves the chicken and egg problem. And this is from the Nobel Prize was given to Sid Altman and Tom Cech. The discovery of catalytic properties in RNA also gives us a new insight into the way in which biological processes once began on this earth, billions of years ago. Researchers have wondered which were the first biological molecules. How could life begin if the DNA molecules of the genetic code can only be reproduced and deciphered with the aid of protein enzymes? And proteins can only be produced by means of genetic information from DNA. Which came first, the chicken or the egg? Sid Altman and Tom Cech have now found the missing link. Probably it was the RNA molecule that came first. This molecule has the properties needed by an original biomolecule because it is capable of being both the genetic code and an enzyme at one and the same time. Okay, and the way it works is, let's first see about DNA. Okay. Well, DNA is this double-stranded helix. Goes from 5' to 3', and reverse goes 5' to 3', and is hybridized, such that this is basically a review of, though going in a little more detail. A and T, this is 2 hydrogen bonds, and G and C are 3 hydrogen bonds, which all make sense. When they come and hybridize, these things give you an energy. The minus signs of the phosphates tend to repel. So actually, what does this say about salt? Let's say you have a certain salt concentration, and like 0 salt, and in one case you have a fair amount of salt in solution. And you throw in some DNA of some length. Is it going to be hybridized if it's in one solution versus another? Okay, you have 1 minute to give me the answer. See, I wanted to see who is actually awake. No, and thank you very much. Okay, so you have strand here, strand here. And let's say it's, let's say 15 bases long. And in one case, you put it in a solution that has 0 salt, no salt. In the other case, you put it in a solution which has a lot of salt, let's say 150 millimolar, which is actually the salt concentration that you typically have in a cell. Which is more likely to hybridize? Okay? You have 1 minute to answer. Quick, go talk to your partner. Go ahead. Go ahead. Talk. Go. You have it? Okay, 1 minute. So who has an answer to the question? Oh, yeah, no, you have, come on. Somebody who has never answered the question. I know this is like, come on. Even if you don't really know. You haven't?
How's the one with salt?
What's that?
The one with salt?
The one with salt makes it what?
More, I don't know, what term do you use?
Hybridized?
Hybridized?
Yeah. Is that true?
Yeah.
Okay, yeah. What? Just hold on 1 second. Why?
I have no idea. You said that was inside the cell, so it makes sense. Right?
Yes.
The positive charges of the cation of the salt might help neutralize the strong negative charges of the phosphate.
Exactly. You have a string of negative ions and a string of negative ions. To get them close together, if you have no salt, there's a lot of repulsion. So a lot of repulsion, and there's a certain amount of attraction. If you add salt, they come around and they effectively neutralize this charge, and so it's more likely for them to come together. Yeah, something quick?
What did you mean by come together? Is that the same thing as hybridization? Like
Yes.
So basically what you meant is like the DNA just twists. That's
Well, it's not a matter of twist, but forget the fact that it twists. This is hybridized, and this is unhybridized.
Oh,
Okay?
okay. So the salts neutralize the negative charge, so the hydrogen bonds can take over.
Exactly. There we go. Okay. Now a small.
Does that mean that DNA always has salts attached to them?
Yeah, indeed. Yes. Oh, essentially always. Of course, it depends on how much salt there is. Okay. Now, if the thing was RNA, what's the difference? The difference is deoxyribose. Deoxy means that this is just an H at what's called the 2 position. For RNA, it has an OH there. So it seems a small difference, but it's a huge impact. Another factor is that you have thymine over here. Thymine has a methyl group right here. In RNA, it's substituted for a uracil, which is basically the same thing, but without the methyl group. And it turns out that again, it has U instead of T. We're RNA. However, U is not as good as T. Why not? Well, because U can hybridize actually to G and other things, right? You have A and T. They're very particular. Where if you have U, it is not as particular. So you can possibly have more mutations involved. So thymidine greatly improves the efficiency of RNA, of DNA replication. Also, it turns out that methylation in thymine protects the DNA from viruses. And in general, RNA is single-stranded because this OH is adding a little bit of bulkiness, though in fact RNA is, again, mostly single-stranded, but also it can form double-stranded. It can wrap around itself. And this adds to chemical diversity, which is good if you want things to react. That is, if you have something maybe in double-stranded in one situation and then it goes into single-stranded in another. Okay, but more, the big news is that RNA can self-cleave. That is, you want to have an OH or an oxygen here. It turns out that in basic conditions, you typically have your O-, and it will come and attack the phosphate over here, which will cause this oxygen to leave, and it breaks apart. The fact that it can self-catalyze, meaning it can do a catalysis, is critical. And so basically RNA can be its own enzyme. In fact, they're called ribozymes. That is, it's an RNA plus an enzyme. And it turns out that well, first it's a property that DNA doesn't have. So it finally breaks the problem of how does RNA produce anything by itself. And furthermore, what has happened is this property of RNA is still used today. Remember when you have the ribosome, we have DNA to RNA to protein, and the ribosome is there to bring the RNA to the protein. It turns out that the ribosome has RNA in it. It has protein, but also RNA. And it's actually the RNA which does the enzymatic coupling between RNA and the protein. Okay, and there's, a nice website. Okay, what we're going to do is we're going to take a 2-minute break, and then we have less time than I originally thought, but we will go at least and begin the new topic of X-ray crystallography and how to determine the atomic structure, in this case, of DNA. Okay, so it's 10:33. How about till 10:35? So I want you to stand up, stretch your legs, and if you want, some people can ask me questions. Get a drink of water.
Will there be office hours for like homework help and stuff tomorrow?
Yes.
I know there's an office hour after class today, but I'm not really
Yeah.
available.
Yes. Josh? She's wondering about, there's an office hour in terms that they have homework due on Thursday, so that
It will be on Wednesday afternoon.
yeah.
Or, sorry, Wednesday morning. Mine's in the afternoon. That's why I think it's all in the afternoon.
You have your like biophysics.
Mine's on Friday.
But we have two other TAs, and so Wednesday at what time?
I'm gonna walk down to Cindy's office because she has it on the door.
Great.
I don't remember.
Great. Why don't you walk down and?
Is there some other way to get help?
Yes, although we have to figure out what that is. Email me right away and we'll work something out. What time?
How they structure it, you have to memorize everything. I know. You have to know everything.
Josh, you said 9:30 to 11? 9. What is the room?
So it's 179.
Why are you doing that? Aren't you supposed to take that too?
Yeah, but I haven't taken it yet.
Yeah. I took that TV like
Okay, there are office hours tomorrow, Wednesday, from 9 to 10:30. They're in 179 Loomis Lab of Physics. If you can't make that, that we do also have it on Fridays. I know many of you do not schedule things that far in advance, but if you're dying to meet with a TA, we'll try to have something. So email me right away, though. I don't want a TA to show up and then, no one shows up. Okay, so we'll have about 12 minutes of this, and this is X-ray diffraction, and that is one of the premier techniques of structural biology. And I want to emphasize it's truly because of physicists going into biology that this technique exists. And in fact, nowadays, there is another technique which we'll study in a little bit, and that is electron microscopy, which is, again, it's becoming the premier technique. It's giving a good run for its money on X-ray diffraction, in particular what's called cryo-EM, which is making the things very cold. So for those of you who are physicists, this is a great example of showing how you can use the physics ideas for new techniques which will be ultimately really important in biology. Okay, and the first one that was ever done was whale myoglobin, fairly small. much more recently, which also won 3 Nobel Prizes, was the ribosome. Also, I told you the last time they've done the spliceosome. Okay. This is, of course, the structure of DNA. In particular, what we're going to do is we're going to figure out the periodicity of this. We have the interbase spacing, which is 0.34 nanometers or 3.4 nanometers. We're going to find that it has both a minor groove and a major groove. So how can we do this? And the idea is X-ray diffraction. You have an X-ray beam. Typically, you have like a copper, and you basically heat it up and you can get some X-rays coming out. Nowadays, in fact, you have things like the advanced light source where they have rings, and that, it turns out, you can get much better X-ray diffraction out of. Anyways, and you have to make a crystal out of it such that it is not moving. And this will diffract. And then you need to collect the diffraction pattern. And then what you need is you have the diffraction pattern, which is basically what I showed you in terms of DNA. Then you need to back transform. And generally, actually, it's using Fourier transforms to go back to the original structure. So the maximum possible resolution is on the order of lambda. Lambda is the wavelength of light that you're hitting it. Why is that? Yeah. Say it again. The uncertainty principle. Okay, explain that to me.
So there's a limit of the resolution. So I don't know the formula about that. Do not know how to derive it in a simple
Okay, we're going to actually see if we come up with a simple formulation of that. But that's right. Initially, you know, we want to see like the 3.4 angstroms between one base pair and another base pair. So you know we're going to need at least 3.4 angstrom radiation to be hitting and to diffract. Now, what if you hit the DNA with something much longer than 3.4 angstroms? Will you see diffraction? Not so sure. Well, just wait a minute. By the way, this X-ray diffraction is an elastic scattering. That is, you have the DNA and you have the light, and you get elastic scattering. You don't, what's elastic scattering as opposed to inelastic scattering? Who can tell me? Yes?
Energy conserved.
Okay, energy conserved, yeah. Go ahead. Yeah, what does that mean?
Well, typically for experiments like this, scattering, elastic scattering, you pick the same wavelength
as you put incident on the sample.
Okay, exactly. You have wavelength, and it comes and it scatters. What does scattering mean? Bounces, doesn't like keep going, but it goes off at some angle. And in fact, that the wavelength that you're detecting is the same as the wavelength that you're shining into it. You're not introducing you're not putting energy into. Okay, so this is the famous picture. So what we need to do is we need to understand first that it's clearly in X. How do you get that? What else do we need to know? That there's layer lines, like 0, 1, 2, 3, 4 seems to be missing, and then 5 and 6 and so forth. And this distance is going to correspond to some distance within either maybe this distance or this distance. And in fact, that we have 10 of these layer lines. What does 10 mean? We have the missing 4th layer. Why is it missing?
Distinguishes the major and minor.
What's that?
Does it distinguish the major and minor?
That's an interesting question. That is, does it distinguish between the major and minor groove? I don't know. Let's quantitatively figure this out. Yes?
Something like in there, for instance, the 9th and 8th, 9th, and 10th layer. So how do you distinguish the layers? I mean, everything is black. You cannot layer them.
Well, here you can clearly layer them, right? And it's true.
How do you understand? I say it's 11.
Yeah. I don't know. I don't know. So, whip me, or I don't know. Yes?
I guess I'm a bit confused as to what you mean by the major and minor groove. Like, what does that refer to?
Ah, good. Oh. Okay. This is the DNA. So it has who can explain this better than I can? No, you're a TA, of course. Yeah, okay.
So from what I know, the nucleotides are aligned in so if you have the DNA strand, instead of it being like perpendicular, it's like 120 degrees or something.
Yeah.
And so that kind of like creates a bigger gap on one side than the other one.
Yeah. Yeah.
I think that's because the DNA is twisted. So my fourth base pair is twisted then, and not the thread.
Okay. Let me give it a try. So you have one. Just imagine a string. And then you have another one, which you're going to have exactly the same one. And you put it over here. Okay? Now, let's say you have this, and now you're going to take it and you're going to have like this. Again, these are exactly the same as these. It just differs of the phase between them, right? So in this case, there's no major and minor groove. Here, however, there's a minor groove and a major groove. And this, it turns out, is the structure of DNA. And this structure is, of course, determined ultimately by the angle of the base pairs. And so, in fact, this is the DNA, and it has a major and a minor groove. Okay? Okay.